What could this possibly be?
Those are tough. On the bottom, 0.1 points.
Yes, this is such a hard.
I was losing.
Probably not.
I lost the whole game.
Any questions before we get started?
You broke it.
You looked at the multiple choice yet?
Yeah, send them out.
Oh, you did?
They're posted. I think.
Multiple
What's that?
I like it.
Seem good.
What did you say?
I think so.
Why would you want to? Actually, that's probably better than mine messed up.
All right, well, if there's no questions, we'll get into it. Back to the grind. Why can't every day be the same day? Okay, so the first part of the class was just about stuff that we're going to see time and time again. And now we're going to kind of work our way through the nervous system. We're going to start kind of on the outside and talk about nerves. You'll tell us a bit more about specific peripheral nerves on Monday. That should be fairly brief, all the talks. It's just one nerve, sensory distribution, motor function, what it looks like when it doesn't work. And we're going to work our way inward from there. We'll hit the spinal cord, talk about the tracts in the spinal cord, a little bit about the autonomic nervous system, and then we'll have an exam. We'll move into the brainstem, cranial nerves, then we'll have an exam, and then the cerebrum. And we'll talk more about maybe some higher order functions like motion and memory, and then we'll have an exam. But the same ideas are going to come up time and time again. You're going to hear about neurotransmitters. You should probably know the different receptors that they have, and what coupling is, because you'll hear that over and over and over. Those are just kind of the basic building blocks of the nervous system. There's always going to be some neuron involved. It's going to spit something out. It's going to bind to a receptor, and some effect will occur. So we got that now. Now we can talk about system functions. There's going to be a little bit of cells and molecules here. Not going to get into pathways yet. We're going to smoothly transition. And smooth transitions are important to the nervous system and to muscles as well. A lot more on that in the next lecture. But we don't want jerky movements. We don't want abrupt changes. So we're going to gently coast away from cells and molecules into systems. We'll touch on axon transport again. That's fairly important for how our peripheral nervous system works, because we're not making any proteins out here, at least as far as our nerves are concerned. We have to transport it there. And when things wear out, which they inevitably do, we have to take them back to the cell body and recycle them. So we're going to be moving stuff along axons, and sometimes there's considerable distances to traverse. Because of their great length, axons are also sites of injury, so we'll review that again. If you remember last semester, then you're good to go. And if you don't, we'll go through it again, that's all right. And in between there, we'll talk just a bit about the very basic structure of a peripheral nerve. Because it's not just a bundle of axons. It's mostly a bundle of axons, but there's a couple other things in there that matter. Hold on. This is either a really bad sign or okay. Oh, okay. Could be a really bad sign. Could be. We'll see. Who knows? Okay, great. Great. This is wonderful. You guys didn't want to be efficient today, did you?
No.
Okay. Good. Yeah, fine. There was this. All right, we'll try that one more time. thank God I had that wonderful transition. Man, things were just flowing. This would be one of those abrupt stops. One of those abrupt changes that we don't like. Oh, there we go. Oh, great. All right. Okay, we're back in it. All right. So, let's not forget that we're only making proteins in our cell body, a little bit in the dendrites, but not in the axons. But we still do stuff in the axons, and we still need proteins. What do proteins do for us? Thank you. Thank you. Good. So we're all on the same page here. Now, the way that molecules find their way down the axon is, of course, by riding along the cytoskeleton. How else are they going to know? You know, they don't have eyes or anything like that, so they need something to guide them. And when we're talking long-distance transport, we should be thinking microtubules. We shouldn't be thinking neurofilaments. These are nice and all, but there's no polarity to the subunits that make them up. In other words, there's no directionality to them. This way and that way, they all look the same. Not true for microtubules and microfilaments. The proteins that make them up have a sidedness. They have plus end, minus ends, and that allows molecules that latch onto them to know, am I moving toward the center of the cell or am I moving away from the center of the cell? Here we can see the center of the cell. Those yellow filaments coming out will be the microtubules. And notice they're all organized in a single microtubule organizing center. That's how things know where the center is. The nucleus is usually pretty close to this. So after we're freshly made proteins, if we want to move to the periphery, just move toward the plus end of the microtubule, and that'll get you there. Toward the edge of the cell, we can see our kind of purplish actin filaments. These are going to be for more local trafficking. Once we get in the general ballpark, by riding along the microtubule express, we'll hop off and get onto a microfilament to hit our final destination. Transport in the dendrites is chaos, so we're not going to talk about that. We're going to talk about transport in the axon, though, because that's what we're extending from the spinal cord out into the periphery. The reason it's not so chaotic in the axon is because we actually have nice organization of our microtubules. Plus end is out here, minus end is over here. So we can tell which way we're moving. That allows motor proteins like kinesin, which moves toward the plus end always, and dynein, which moves toward the minus end always, to carry out anterograde and retrograde transport, respectively. So if you're a newly made protein, you want to get down to the synapse, hop on kinesin, because that's going to attach to microtubules and run toward the plus end. If you're an old protein, you've got a few miles on you, you're not functioning properly, hop onto dynein. That's going to run toward the minus end and bring you back to the cell body where you can be inserted into a lysosome and broken down. So we got two directions that we're moving, anterograde and retrograde. Your ultimate destination is going to be determined by, of course, myosin-dependent protein on microfilaments, because those are what we find out there in the periphery. At synapses, these are the filaments that give the synapse its actual structure. The shape of the cell is going to be determined by microfilaments because they're very dynamic, as we'll see in the third part of this lecture. What is this? What part of the cell am I looking at here?
Dendritic spine.
Axon, dendrite, yeah, dendritic spines. All these yellow things coming off, those are those little protrusions that come off of dendrites. They're very dynamic synapses. The reason that they're yellow here is because they're filled up with actin. Here we can see a cartoon schematic showing us our postsynaptic density. It's dense because it's full of proteins. Postsynaptic because it's postsynaptic. And immediately below that postsynaptic density, we have an actin network. This can rearrange, it can grow in size if we want to strengthen that synapse. Maybe it's meaningful. Maybe it's not meaningful and we want to shrink it away. We're always treadmilling our actin filaments. So if we don't put energy into maintaining this, it will retract itself. Actin is very dynamic. Microtubules are far more stable. This is going to be important whenever we're looking at axon regrowth. But that long distance transport is all about microtubules. Microfilaments are more about dynamic structures. The microtubules are going to stay there for you all day so that we can run along them in the positive direction for anterograde transport. And this moves at different paces. On the bottom here, we can actually see anterograde. There's fast anterograde. You see that one zip by? There goes another one. Look at these little red blobs moving around. These are neurotrophin receptors. One specific type called TrkA, which is going to bind nerve growth factor, which is exactly what it sounds like. We can see here that these receptors are moving away from the cell body, so that's anterograde, or back toward the cell body, that would be retrograde. And what you'll notice here is that some of them are moving very quickly, some not so much. And that's because the stability of that interaction with kinesin isn't constant. This is still chaos. We're still talking about molecular function, so it still comes down to chance. Do you bump into kinesin and the adaptive proteins that will hold you there? Do you happen to fall off? How frequently do you fall off? That's what's going to determine your rate. Some things tend not to fall off very often. Synaptic vesicles, neurotransmitter precursors, mitochondria, things that we need to get down to replenish our synapses. Hell, a fresh batch of lipids, you're going to need those every once in a while. Sometimes we cut those up. We need to get fresh ones down there. Proteins that we embed in the presynaptic membrane. Snares, for example. We have to replenish those when those wear out. We want to get those there quickly. So those interact stably with kinesin, and thus the chaos, that random diffusion, now has direction to it. Because they stick on kinesin, and kinesin runs toward the plus N. Others don't interact quite as stably, and they move more slowly. You can break this slow transport down into the slow component A and B if you want to say that the cytoskeletal components will get there a little bit slower than our soluble proteins. That's fine too. Or you can just say fast and slow, and you can notice that these are fairly different from each other. We might be moving up to, I don't know, half a meter if you're lucky in a day. That's pretty good compared to maybe half a millimeter in a day. So orders of magnitude different here between fast and slow. And that's all because different proteins interact with different stability. This is a more modern day approach to seeing this. And you can see both retrograde and anterograde transport. Here's how we originally figured all this out. This is a pulse chase experiment where you put on some radioactive amino acids, do it in a few hours. These will get incorporated into newly made proteins and shuttle down the axon. And those that move quickly are going to get there sooner. And those that are slower will get there later. And you can just watch the movement of radioactivity down the axon. So they only deliver a single pulse of radioactivity, so they know for 15 minutes we gave it radioactive amino acids, then they were washed away. So any of this radiation that ends up down the axon, which we can measure across time, must have come originally from the cell body. So they could see actually 2 or 3 distinct pools moving along the axon. And the reason for that is because of different interaction with this motor protein here. Kinesin is always running down the microtubules. The question is, does it have any cargo? With these cargo, it's very likely. And that's why they're transported rapidly. Less likely down here, but we'll still get net diffusion in the plus N direction. Retrograde is just going to be from a different motor protein. Dynein runs toward the minus end, therefore it's going to move toward the cell body. There's two things that we're doing with retrograde transport. One is removing old cell components, like proteins or organelles, because we still have mitochondria down there in the presynaptic site. We're still going to generate free radicals. Those are still going to donate electrons and break apart macromolecules. And when that happens, we need to get rid of them. A protein that's not functioning properly is of no use. So we'll hop on dynein and move back to the cell body. We'll put these damaged components into lysosomes, break them apart, and recycle the building blocks of them. The other thing that we do is provide some sort of feedback to the nucleus about how active this neuron is. How often are we communicating with our postsynaptic target, for example? Have you ever heard use it or lose it? Well, it's true. When a neuron communicates with a muscle, that muscle provides a little feedback. That feedback comes in the form of nerve growth factor, for example, so neurotrophins that bind to those track receptors that we just saw zipping on the last slide. Well, when those are stimulated, they head back to the nucleus and stimulate the expression of pro-survival genes. They keep the neuron from killing itself. Because that is the logic of the nervous system. Make an excess of neurons. Those that find their target will get neurotrophins and they won't kill themselves. Those that don't find their target and innervate a muscle, they should kill themselves. They're not doing anything for us and they're just wasting our ATP. They're wasting our glucose and our oxygen. Get rid of them. Only those neurons that we're actually using should survive. And that's why we use it or lose it. So if you're not innervating the muscle, if you're not receiving that positive feedback, well, now we're going to send a different type of receptor back that's going to activate pro-death pathways and cause the neuron to be eliminated. So that retrograde signaling is very important for kind of fine-tuning our nervous system after development. We have this excess, and we need to get rid of it. The way that we do that is just keep the ones that actually innervate a muscle alive. How do we know if they innervate a muscle? There's only one way. We have to have feedback from that muscle. And we got to take it all the way from the periphery back to the central nervous system. That's, of course, accomplished by dynein. Now, the rate here is somewhere in between the very fast and the slow component of retrograde transport, as you can see. And we want this to be fairly quick. There's an important task here. Keep the cell alive. Life and death decisions are made based on retrograde transport. That's not enough to get you excited. I don't know what it is. Maybe some toxins? This is the bad side of that. Sometimes toxins can latch onto those receptors that are going to hop onto dynein and move back to the cell body. This is how we can get some viral infections like rabies. We get tetanus toxin infection as a result. That can lead to paralysis because of uncontrolled muscle contractions. Maybe that gets you going, I don't know. But what's going on out there in the periphery, here's our presynaptic site, here's a little receptor that we've activated because we talked to a muscle, and the muscle said, here, take this and live. We take it into the cell. There's a few ways that we could endocytose it. If you want to talk about all those, we can. But either way, we pinch off a vesicle. There's a whole bunch of proteins involved here, which we're not going to talk about. The one we care about is this one, dynein. Dynein is the motor protein right here. We're going to attach via an adapter to this activated receptor, and we're going to run this whole complex back to the cell body so we can affect gene expression. And then our neurons live, and we live. Or we can show up to class and learn. I know. Do you have any questions? I also accept feedback. Okay, well then let's review these and we'll move on. So that interface between spinal cord and skin or muscle or tendon, that's going to be through our peripheral nerves. The input, the output. So we're going to be talking about big bundles of axons here. Now when you're in the peripheral nerve, it's a mixture. You're going to have sensory and motor components. They're going to be all mixed together, so that when you have peripheral nerve damage, you're going to have sensory and motor disturbances. There's a separation once we hit the central nervous system. More on that in lectures 9 and 10. But for the rest of these signaling pathways, motor and sensory remain separated as we move up the spinal cord into the thalamus and eventually to the cortex to produce some sort of perception. Or when we're going from the cortex down to the spinal cord to carry out a movement. They're kept separate. In the nerves, they're mixed, and that mixing occurs as they exit the vertebra there. That's where our dorsal portion here, the sensory component, which contains afferents, so those that are arriving into the spinal cord. Information flow is this direction. That's where those sensory afferents are going to meet up with the motor efferents. They're exiting the spinal cord to cause some sort of effect. Whatever helps you remember the A and the E, use it. Some of those effects are going to be just muscle contractions, overt changes in body position. And then other functions could be smooth muscle contractions, changes in heart rate, things that the autonomic nervous system carries out. Once they come together here, now we're mixed. And we're going to have both sensory and motor components. Before we exit completely, we send off a little offshoot to innervate meninges there, ligaments, intervertebral discs. So we're innervating the spine itself here so that whenever we have damage, movement of discs, inflammation, we can pick that up. We can detect that damage and we can feel back pain as a result. So this meningeal branch is a big source of back pain. Anytime you have spinal abnormalities, meningeal branches get stimulated and they create the perception of back pain. You often don't see these. You don't see them here in this cartoon, but they're fairly important because back pain can shut you down. Once we exit the spinal cord, whenever we're in regions that contain the autonomic efferents, we have to meet up with a postganglionic neuron. For the sympathetic nervous system, that's going to be in your thoracic and upper lumbar regions of the spine. For parasympathetic, they'll just be down there in the sacral portions. But you won't have these sympathetic chain ganglia there. It'll just be some ganglia near the target organ. That preganglionic axon that exits, so our vertebra is here, here's that intervertebral foramen, so we've exited and now we're outside the spine itself. Here's this chain of postganglionic neurons that runs alongside the spine on both sides. That myelinated fiber comes around through the white communicating ramus and then heads back as an unmyelinated fiber from the postganglionic neuron. So your autonomic signals are going to be a little bit slower because of that lack of myelin. Just keep that in mind. That's okay because what they're innervating acts slowly anyway. So you're not producing some rapid response to environmental stimulus like you are with just somatic efferents. There we want immediate movements because we might need to get out of the way of danger. Slowly adjusting so that you're ready to have a meal and digest, we can take our time with that. So there might be this little loop here so we can communicate with a postganglionic sympathetic target, and that's only for the autonomic nervous system here that we'd have this. For those postganglionic fibers and everything else that exits, we're then going to divide and split into a smaller dorsal component, so the dorsal rami, and then the larger ventral component. The dorsal component is really just going to innervate the back. muscles, skin. It'll hit the internal organs as well. The ventral component is much larger because it's going to hit a greater number of targets. It's going to hit the limbs, neck, front of the trunk. But these ventral branches are going to be much larger just because they have a larger territory to innervate. Whether you're in a dorsal or ventral component, doesn't matter. The peripheral nerves are all going to look pretty much the same. They're going to basically be bundles of axons with a few extra components. This should look very familiar here. Now, of course, we want to protect those axons. They're not encased in any bony structures, and so they're liable to be damaged from time to time. You're going to bump into things. So we want to provide some cushion. We have a few layers of protection. The outer component would be the tough epineurium. This is the dura mater, just extended out onto the peripheral nerve. Same thing though. Surrounding these bundles of axons, we have a continuation of the arachnoid and pia mater, called the perineurium. This is going to prevent any diffusion of toxic components in those blood vessels, the large blood vessels found throughout peripheral nerves, from diffusing in and damaging axons. So this separates the nerves themselves from that connective tissue around there. So this acts as a diffusion barrier to keep these axons isolated from the blood. Blood is toxic to neurons, so we want to have a few diffusion barriers built in. This is kind of like the blood-brain barrier, except it's out in the nerve. Within these bundles, we have sort of a kind of like gelatin in a way. It's collagen, so it's just a very tight gelatin. But a bunch of collagen fibers that create essentially this gelatinous matrix called endoneurium. So it's a cushion that all of these axons, it's all these little circles here, and the glia found within them, they're going to float around in this gel. Again, to provide a cushion to make sure that there's no extreme pressure placed on our nerves. Because that pressure is going to first kill the glia, then kill the axon. Sometimes that happens, we'll get to that in a bit. So here's another cross section. We can see the larger blood vessels here. Outside are bundles of axons, those fascicles that are surrounded by perineurium. The glia in there are going to be Schwann cells. They're always Schwann cells, but not every Schwann cell is a myelinating Schwann cell. Of course, when you learn about oligodendrocytes and Schwann cells, you learn that they make myelin, and that's totally true, except when it's not. There are non-myelinating versions of both. And those non-myelinating versions of both are analogous to astrocytes in terms of their function. They're going to buffer, in this case, potassium, because we're not forming synapses within the nerve itself. All we're doing is conducting action potentials. We don't need to pick up any neurotransmitter here. We're not releasing it. But we are releasing potassium. What would happen if we didn't buffer that potassium? Let's say extracellular potassium levels rise. Then what? Annie, happy birthday. And what would happen if we didn't buffer that potassium? What if we didn't clean it up? So we conduct an action potential down the axon. Sodium rushes in, potassium rushes out. If we don't soak up that potassium, what happens to extracellular potassium levels?
You get way too high.
There you go. What happens to the reversal of potassium?
Kind of brain dumped after that test, to be honest.
I see. I see. So we have less of a diffusive force. Do we need a strong of an electrical force to offset that? No. So we're not going to be as negative. We're going to depolarize. What are we most permeable to at rest? Oh, there you go. So the cell is going to depolarize. We don't want that. We want to make sure that we keep extracellular potassium levels low so that the action potentials are only generated in response to some sort of input, either from the periphery if it's sensory, or from the central nervous system if it's a motor axon. We need to keep potassium low so that our activity occurs during appropriate times. When this doesn't happen, the smallest fibers are the ones that are going to get activated, and those are the ones that conduct pain. You don't want this to happen. I believe we'll talk about this when we discuss the trigeminal nerve. But this happens on occasion. When these non-myelinating Schwann cells are damaged and they don't suck up potassium, the action potential can actually jump from one axon to another. And the axons that get excited by that extracellular potassium are the ones with the highest input resistance, because V equals IR. That's still true, by the way. So the small pain-sensing axons will get stimulated, and you will feel pain, even though there's nothing actually damaging your tissue. So myelin's great, but so is potassium buffering. These Schwann cells are also going to do a little bit more than myelinate. They're going to provide, they're going to be the middleman between blood and axon. They're going to shuttle glucose, for example, into neurons so that they can pay the bill. They can keep their sodium-potassium pump going, so we maintain that diffusive force for all of our ions, so we maintain proper resting membrane potential. And just like astrocytes, these form a big network. They have gap junctions that hold them all together. Now I know it looks like this non-myelinating Schwann cell here is myelinating these little axons, but it's not true. It's just simply surrounding them, but it's not removing ion channels from the axon itself. It's just sitting very close so that whenever the action potential occurs, we can soak up that extracellular potassium. But non-myelinating Schwann cells are still important. They're also going to play a role in injury recovery, but that is a topic for the next part of this talk. Before we go into that, do we have any questions? Alrighty, then let's work through these. We'll move on to the last part. Normally those work pretty well. You can live your life and often not have significant nerve damage. But on occasion, nerves get damaged. It could be a nerve, it could be a tract in the central nervous system. That's far worse, because the peripheral and central nervous systems are going to recover differently. This is still true. Axons are, of course, the primary site of injury.
She Googled it.
I just
Tomato
Googled
jello?
noodles and jello.
Huh.
Continue.
So those noodles are very long. Therefore, they occupy a larger space than other components of the neuron, and therefore it's just more likely that axons are going to be injured than other parts of the neuron. They're spread out over a larger area. So by chance, you'd expect axons to be injured. This is true whether we're talking central or peripheral damage. It's especially true in the periphery because it's just a bunch of axons. But even in the central nervous system, axon loss occurs whenever we have nervous system injury. They are very easy to break because they're long and thin. And here we can see the loss of axons. Sort of. We're actually just comparing two different people. This is not a before and after. And different folks have different numbers of axons, but bear with me. All of these different colors are just telling you kind of the direction that water tends to diffuse. Water has directional diffusion when there are barriers to its ability to diffuse. It should, like everything else, just spread out in all directions. It should be random. It's not, though, whenever that water is moving within a bundle of axons, because of the myelin. It's a bunch of fat, and fat and water don't mix. So when the water is surrounded by bundles of axons all moving in one direction, well, the water can really only move in that direction. And we can pick that up with MRI and trace our axons. That's what we're looking at here and here. And you'll notice there are some significant gaps in those axon tracts. Normally we should have our axons radiating out from all of the corpus callosum here. So this is the bundle of axons that's connecting both sides of our cerebral hemispheres. And we should be connecting all of the cerebral hemispheres, essentially. We bias more toward the frontal lobes, but we're still going to hit all the lobes. Notice it's nice and fairly uniform over here. Now, we've had some injury, and so axons are lost. When this occurs, when axons are damaged, there's going to be degeneration away from the cell body, because this can't make any new proteins. It can't do anything. It has a limited supply of what's there. It can't make any new proteins. It can't respond to that injury because it doesn't have a nucleus. It can't actually change gene expression. It's totally dependent on the cell body in order to replenish the proteins that are there. Now it's been cut off. When that happens, this is going to wither. It's going to wither because the glia there are going to destroy it. It's going to also destroy itself. We're going to cut it up into little bits, and we'll eat that and recycle it. Everything is fine. But this neuron here is going to have to regrow and retrace that lost axon. You might see a little bit of retrograde degeneration, but for the most part what you find is anterograde degeneration. So away from the cell body. And that should make good sense. Only the part that can make proteins will survive. Now out there in the periphery, axons can regenerate fairly well, and that's because of the Schwann cells that are there. They're going to play a few key roles in allowing axons to regenerate. One of the things that they'll do is clean up the mess. They'll spit out proteases. Proteases are just enzymes. They cut up proteins. So they're going to start to break apart that cell debris that's there and make it a little more digestible to the macrophages that they recruit. We're not in the central nervous system anymore. We have access to the immune system, so we can recruit them. Schwann cells are going to help do that. So they'll clean up the mess themselves to some degree, but they'll also bring in the pros, the macrophages that are designed to engulf and recycle damaged tissue. So here we're seeing a little crush injury on an axon, so when you squeeze it, that's going to cause it to pop and die. We have our anterograde degeneration. Part of that's driven by the Schwann cell here, and it's also going to be by recruiting immune cells that are going to eat up all of those little bits of axon and clear the way. So they remove the debris. That's your anterograde degeneration. We're getting rid of the rest of that axon, but not the proximal portion, because we're going to try to guide this to its target. Schwann cells are going to help do this. What they'll do is secrete guidance cues and neurotrophic factors that are going to keep that axon alive and stimulate growth of the cytoskeleton. That's what guidance cues do. There's really two things that they can do. The first is allow the cytoskeleton to grow. The other thing that they can do is allow the cell to stick. And both of those really need to happen. We'll see more in just a bit on that. And Schwann cells will do both. They'll keep the cell alive, tell it to create a greater amount of protein. It's going to need to because it has to actually grow. It's going to allow that growth by stabilizing those cytoskeletal filaments so they elongate. Now we're biasing the cytoskeleton toward growth rather than an equal amount of growth and retraction. Cells don't grow because they're destroying their cytoskeleton at the same rate that they're building it. But they don't sit there and do nothing. They're always just treadmilling their cytoskeletal filaments. They're pulling in and growing more out. It might look like nothing, there's just no net movement, kind of like a reversal potential. Ions still move. When cells aren't growing, their cytoskeleton is still moving. There's just no net movement. We want net movement, so we stabilize those filaments so they don't fall apart. And then we give the axon something to hold on to. If it can't actually put its foot down and take a step, it's just going to be flopping and it won't stick. Why does an axon end up in one place versus another? It gets in where it fits in. Wherever it can bind, it sticks. And if it can't bind over here, it won't go there. It's just that simple. We'll see in just a moment. Once this is all said and done, and we've regrown, we've followed along in our Schwann cell here. Here's that growing portion of the axon. It's called the growth cone. It's kind of cone-shaped, not really, and it grows. So that part is pretty good. That's going to grow along to retrace the original path of that axon. If it was myelinated, we'll remyelinate it. If it wasn't, we won't. When we keep our connective tissue intact, such as in a mild crush injury, for example, rather than a laceration that actually severs the nerve, regrowth is a lot more probable because we have this nice little road here. We haven't severed our fascicle bundle. We still have the road. We still have that path of Schwann cells that we can step on, one step at a time, to reach our target. So here's a growth cone. Here's what's actually going on whenever neurons are growing. There's two components here that we can see. And we'll get it moving, and we'll get it moving now. The green component is actin. You're going to notice this is very dynamic. It's going to be out toward the periphery. The red is tubulin, microtubules. Those are going to be a little more toward the center, and they're a little more stable. What they did there was apply one of those guidance cues. Notice how we get these long extensions. We've stabilized the cytoskeleton and allowed it to grow, and it's growing toward that. We're biasing cytoskeletal growth toward this guidance cue. That's what Schwann cells are doing. They're allowing that growth cone to extend cytoskeletal filaments so that it moves down toward its target. The other thing that the Schwann cell is going to do is give it something to stick to, because that actin is always getting pulled back. I want you to notice these green filaments. They grow out here and then they get pulled back in. That's always happening. Actin is very dynamic. We're always building it and pulling it back. It's called actin treadmilling. It's like a treadmill. It goes nowhere if the rate of growth and pull is the same. It also goes nowhere if when you pull on it, it's not stuck to anything. This is the other function of Schwann cells. When you send out that filopodia, that little finger-like projection of axon, if you have nothing to grab onto and you pull on it, the cell doesn't move. That's what we can see up there. No movement. We didn't stick. We didn't have a Schwann cell there to guide us. On the other hand, if that actin filament and the surface cell adhesion molecule that it's attached to can stick, for example, if that's a Schwann cell down there, then whenever we pull on the actin, rather than pulling the filopodia back, we pull the whole cell forward. We pull our growth cone in the direction that we want to move. So now, here's our new set point. We grow out another filopodia. Do we stick? If we stick here, we'll go that way. If we don't, then we'll just pull that filopodia back, and we'll send another one out. Do I stick here? Great, I'll go this way. The only way that the growth cone knows where to go is places where it can actually extend its cytoskeleton because it's being stabilized through the release of guidance cues, and places that it sticks to. Because if it doesn't stick, it just pulls back that filopodia. And this allows the axon to slowly crawl its way to its target. And it works fairly well in the periphery. This is not the case in the central nervous system. We have a different set of glia there. Oligodendrocytes and Schwann cells make different myelin. The myelin that Schwann cells make is a little fluffier. Hopefully we remember that. A little more cushion there for whenever you get to pushing on things. You keep your axons intact. Not so in the central nervous system. We have a skull. That's going to protect us, and it also sets the amount of space that we have available for our nervous system. So we need to conserve space. We can't make this big fluffy myelin. We need to make it a little more compact. So we're going to have different proteins that allow that to happen. The different proteins that we find in myelin from oligodendrocytes, from Schwann cells, also affects how axons can grow along that myelin. One of these proteins is no-go. Axons won't grow on it, hence the name. The receptors that no-go binds to are going to destabilize actin. So you can't grow out your little finger-like projections. Here's a nice growth cone. Here's a growth cone. Everywhere a growth cone. But down here, notice, there's no growth cone. We're not forming this collection of little actin filaments that we're sending out because we squirted no-go on it. And we're stimulating no-go receptors and we're destabilizing those filopodia. So we can't try to crawl around in our environment, in part because of destabilized actin filaments in the growth cone. Oligodendrocytes don't want axons to regrow. We don't want to rearrange our central nervous system. Once we put it all together and we learn stuff and we put important information in here, well, we want to make this fairly stable. We'll allow for little subtle changes, but we don't want to dramatically rewire our central nervous system. So oligodendrocytes keep that unlocked by having surface proteins that destabilize the growth cone. No major regrowth of axons. You can see it here, and you can see it in this experiment we saw last semester. It's as old as I am, and it's still true today. They put a bunch of embryonic nerve cells here in the center of a dish, and then past the optic nerve and the sciatic nerve out different ends. Central with oligodendrocytes, peripheral with Schwann cells. What do you think happened? Well, whenever we look, here's the edge of our sciatic nerve. So if we're looking, hold on, we're looking right here in the dish to see whether or not axons came out. They did. Here they are. Here's a bunch of axons that exited from that sciatic nerve. The optic nerve, on the other hand, there's nothing out here. Just dish. That's it. Nothing grew through that optic nerve because it was filled with oligodendrocytes, with their myelin that has things like no-go on it. So you couldn't stabilize your actin cytoskeleton. You get no net growth. There's only treadmilling. And we don't have stuff to stick to like we do on Schwann cells. Peripheral nervous system, things grow pretty well. Central, not so much. The other thing that happens in the central nervous system is the formation of glial scars. This top panel is just showing you nuclei, and they're highlighting the area of crush injury on the right versus the left. So right and left are the same, except they put a little outline here that you don't need. You can see it right here. GFAP is an intermediate filament found in astrocytes. So the red signal right here in part B is just showing you astrocytes. And you'll notice that they line up right around that area of injury and form a glial scar. They will also affect growth cones. Astrocytes also make surface proteins. And what they do is essentially get the axons addicted to the glial scar, so they don't want to leave. They create proteins that interact stably with the growth cone so that it's never able to exit that glial scar. So when we have a crush injury in our spinal cord here, axons that are growing down are going to encounter that glial scar and then never want to leave. Not only do we have oligodendrocytes preventing axon growth, but glial scars also don't help. And this is one area of active interest in terms of spinal cord injury recovery. How do we break up that sticky glial scar that's holding our axons hostage? Any questions? Are we all visualizing actin now? It's always moving back and forth. It's very dynamic, like I said at the beginning. And you can actually see that in real time if you click back a couple of slides. Alrighty, well, let's go through these and we'll call it a class.